First of all, I would like to thank the organizing committee, and in particular, Dr. Beppe Croce, for giving me the opportunity to present my talk today. I share my screen with you. Here we are. In this presentation, I will talk about the antiproliferative activity of hemp extracts having CBD as the main compound. Cannabinoids have been described in the literature since they possess many interesting bioactivity in medicinal chemistry, as you can see from the left of the slide. In relation to their bioactivity against cancer, they have been mainly described in relation with THC, which is responsible for both the analgesic as well as the antiemetic properties of the extract having high content of this compound. Indeed, THC possesses both analgesic as well as psychoactive properties in view of its capacity to bind to CB1 receptors. It is also able to bind to CB2 receptors, which are present in some cells of the immune system. CBD is a non-psychoactive hemp component, which is characterized by a multi-target activity, as shown on the left of the slide, where you can see the complexity of receptors involved in its mechanism of action. For what concerns the relation of CBD with cancer, this picture shows what has been previously described in the literature. Indeed, in this picture, you can see the antiproliferative activity described for CBD against different human cancer cell lines in vitro, as well as also the in vivo models applied up to now. As you can see, the activity described in the literature are mainly related to solid cancers. For what concerns the specific aim of the study, it was to evaluate the antiproliferative activity of hemp extracts having a different phytochemical composition after a careful chemical characterization in order to identify the one having the best profile, which was submitted to a further assessment of the mechanism of action involved in its activity. The varieties taken into consideration in the study were selected in order to have a different chemical composition. Indeed, a CBD type, a CBG type, and a hybrid variety was selected at the beginning of the study. The sample preparation was carried out using ethanol as is the extraction solvent under dynamic maceration, and it was performed both on the raw plant material composed of hemp inflorescences, as well as on the decarboxylated material, which was done in order to convert cannabinoic acids into their neutral counterparts, thus ensuring a higher degree of reproducibility of the extracts. After having completed the extraction procedures, the analysis of cannabinoids in the extracts was carried out using HPLC. In the slide, you can see the conditions applied in the characterization of cannabinoids. In particular, on the right, you can see the UHPLC method coupled with high-resolution mass spectrometry, which was applied in order to obtain a cannabinoid profiling of the extracts, while on the left, you can see the HPLC UV method that was used for the quantification of main cannabinoids. For what concerns the cannabinoid profiling, target metabolomics was carried out using an inclusion list of more than 200 compounds that are all phenolic molecules previously described in the literature related to cannabis sativa. In this way, it was possible to confirm the identity of many compounds in the extracts thanks to their high-resolution mass spectrometry data, which were acquired both in the positive as well as in the negative ion mode. For what concerns the quantification of cannabinoids, it was carried out using HPLC UV. On the right, you can see a representative chromatogram. The blue line is related to the profile of the raw extract, while the red one is related to the decarboxylated one, in order to demonstrate a complete conversion of the acidic precursors into their neutral counterparts. For what concerns quantitative data, the results are shown in the table where they are expressed as milligram per gram. And this confirms that the first variety has a profile having CBD and CBD as the main compound, while the second one was actually characterized by both the CBGA and CBG as the main constituents, and the third one had a balanced composition of the before mentioned components. Having completed the chemical characterization of the extracts, the subsequent step was the assessment of their antiproliferative activity. And to do this, 3 different human cancer cell lines having a different embryological origin were selected in order to have a complete overview of the potential of the hemp extracts. So a cancer cell line having a mesodermal origin was selected together with one of endodermal and one of ectodermal origin. Cell viability was assessed using a specific assay, which is based on the reduction of WST8, shown on the left, in presence of viable cells, which produces a formazan dye that can be detected by means of a spectrophotometric method. On the right of the slide, you can see the dose response curves that were built using all the extracts previously described, both in the raw form as well as in the decarboxylated form. And the assays were carried out both after 24 as well as 48 hours of exposure. The best result in terms of IC50 value was obtained with the decarboxylated extract from the CBD type variety, which produced an IC50 value around 12 microgram per ml on the K562 cells, which are cells of chronic myelogenous leukemia cancer, as you can see in on the right of the slide. To assess the possible effect of the extract against cancer cells having the same embryological origin, the dose response curves were built against other additional 2 cancer cell lines of the same embryological origin. And even if a good response was obtained, the chronic leukemia cancer cells, K562, were still those that responded better to the treatment. The antiproliferative activity was found to be dependent on the chemical composition of the extract. Indeed, the other 2, having a different phytochemical profile, displayed higher IC50 values, as shown from the dose response curves that you can see on the left of the slide. Then the activity of the extract was compared with the one of CBD, being it its main compound. And as you can see on the right, CBD had an IC50 value around 28 micromolar, while the extract had an IC50 value around 16 micromolar expressed as CBD equivalent. And this means that in addition to CBD, other compounds present in the extract can contribute to the overall activity against this cancer cell line by means of synergistic interactions. To assess whether the observed activity in vitro can be really attributed to CBD rather than to its oxidation product, which is a quinone derivative already described in the literature as HU331, which is a well-known anti-cancer agent, cells were treated with both the CBD as well as with the extract, and then UHPLC analysis were carried out using high-resolution mass spectrometry on cell lysates from this cancer cell line. And on the basis of a detailed analysis of the chromatograms and of the fragmentation pathways observed for the peaks, it was possible to finally conclude that the analyte inside the cells was actually CBD and not its oxidation product. It was also interesting to note that the antiproliferative activity of both the CBD and the CBD type hemp extract can be really selective against cancer cells indeed, even when tested at high concentration on PBMCs, which are human peripheral blood mononuclear cells and are non-cancer cells, this treatment did not affect cell viability in comparison with the control. And this is a very interesting outcome when we think about the discovery of new compounds that are active against this pathology. Then live dead viability assay was carried out, and in particular K562 cells were treated with increasing concentration of the CBD type extract overnight, and then they were stained both with calcine and propidium iodide. The calcine is able to penetrate through the membrane of live cells, where it produces a green fluorescence that can be easily detected using fluorescence microscopy, while propidium iodide is able to penetrate through the damaged membrane of dead cells, where it binds to nucleic acids, producing a red fluorescence. As you can see, by increasing the concentration of the extract, there was a parallel increase in the number of dead cells which were stained by propidium iodide. Then apoptosis was taken into consideration as a possible mechanism of action of the extract. To do this, cells were treated overnight with increasing concentration of the extracts, and then a cytofluorimetric analysis was carried out. The results are shown in the figure in comparison with the control. We can focus on the results obtained with the concentration of 20 microgram per ml. Indeed, in comparison with the control, we can see an increased number of cells stained with annexin 5. Annexin 5 is a phospholipid binding protein that is able to bind to the damaged membrane of early apoptotic cells. There is also a slight increase of cells stained with propidium iodide, which indicates a late apoptotic or necrotic stage. The highest concentration tested caused an almost complete death of all cell line population. We know that apoptosis can involve different molecular pathways, which induces the release of CYTOCHROME-C from mitochondria, and finally the activation of caspases, which are responsible for cell death. So in this stage of the study, our interest was addressed at the investigation of the mechanism of action of the extract. To do this, the expression of apoptosis-related proteins, including P53, PCL2, as well as BCL-XL, was evaluated by increasing the extract concentration using Western blotting. As you can see, the treatment with the hemp extract did not affect the expression of these proteins, suggesting that the apoptosis was not actually mediated by the markers taken into consideration. In any case, we were able to confirm the release of CYTOCHROME-C from mitochondria. Also in this case, Western blotting analysis was carried out. And finally, thanks to a collaboration with the National Research Council of Bologna, live imaging kinetic assay was carried out by means of a 48-hour observation of cells treated with D1. And it is possible to see on the figure that the highest activity was demonstrated after 24 hours of treatment with the CBD type hemp extract. After this time point, apoptosis mediated by caspases 3 and 7 was the predominant mechanism that contributed to cell death. Finally, another experiment was carried out in order to evaluate the effect, another possible effect that can be involved in cell death. That is the activity of both the CBD as well as the CBD type hemp extract on cellular membrane properties. To do this, liposomes were built by incorporating both the extract and the CBD. And then their effect on membrane fluctuation was evaluated. In particular, it was possible to demonstrate that both the extract and the pure compound did not affect the lipid composition of the membrane, but rather they were both able to increase the transition, the membrane fluctuation. So in some way, they are able to modify the mechanical properties of the membranes. And this is an aspect that deserves further investigation. So as an overall conclusion of the work, we can say that a CBD type hemp extract was able to produce an interesting IC50 value against chronic myelogenous leukemia cancer cells. The activity can be attributed not only to CBD, but rather to its synergistic interaction with other minor cannabinoids present in the extract. And the mechanism of action of pure compounds is now under investigation, using both proteomic as well as bioinformatic methods. Our final goal is to identify either an ideal composition of a CBD type hemp extract or is a combination of these pure compounds to be used in the treatment of this pathology, even in combination with existing drugs. Finally, I would like to thank all the researchers who participated to this work, and in particular, all my co-workers at my home university, the University of Modena and Reggio Emilia, in particular, Professor Lorenzo Corsi for the pharmacological essays. The research group of Dr. Tassinari and Taglioli at the National Research Council in Bologna for the activity against CASPASE. And finally, last but not least, a special thank to Dr. Alfonso Botto and Fabrizio Fayon, who gave us the opportunity to go ahead with this study using extracts produced by means of an eco-friendly technique, which is supercritical fluid extraction. On the right of the slide, you can see the paper, which contains all the information that I gave you in this talk and much more. Thank you so much for your kind attention, and I will be glad to answer to any question you may have. Thank you.